Hey guys, so this week we are covering nucleic acids. We're going to cover a bunch of stuff. Let's get started. Nucleic acids are not just your DNA and your RNA. That's what we're typically going to think of, but it's definitely not it. And in this class, we're going to talk about some of the other things that we see nucleic acids function for. So like signaling, energy transfer, like your ATP, and that kind of ties in with your redox coenzymes. Those are all nucleic acids, and so we're going to get into it. We're going to start about our information storage molecules. Okay, so big basic thing is your basic unit of DNA and RNA. So that's your nucleotide. So nucleotides have a phosphate here. And if you've taken genetics with me, you may be taking it now, you may have taken it in the past. You're going to see some repeat slides here. I do borrow some of those, but there are a lot of fresh ones too. So there's a little bit of repeat, but there's nothing wrong with hearing things a few times. It's good for you. So nucleotide has some kind of nitrogenous base with a sugar, okay? And that's connected by a glycosidic linkage, okay? So you got this little bond here, glycosidic linkage. And nucleosides have that same bond, but nucleotides, as opposed to nucleosides, so nucleosides are just the base with the sugar. If I change that S to a T and I tack phosphates on here, and DNA and RNA have a nice sugar phosphate backbone with their bases facing inside. We're going to look at that structure in just a second. So that's an important concept, though, to understand the difference between a nucleoside, no phosphate, and a nucleotide has phosphates. Okay, either one phosphate, that's what you're going to typically encounter for DNA and RNA, but you can tack on more of those, and these are highly energetic. Phosphate is a really interesting atom as an element. It can sort of hold on to more electrons than it should, than you would typically predict. And so you can store extra energy into phosphates. And so that's why ATP, adenosine triphosphate, is a nucleotide. So the structure of nucleotides, DNA and RNA are very similar structurally. So they have, we tend to label them by their carbons on the ribose here. So we've got our 1, 2, 3, 4, 5 carbon. Yeah, that's the 5th carbon right there. And, sorry. So we give directionality to our nucleotides based on those carbons, okay? And so if you can imagine, these are chains of DNA or RNA. In this case, it's DNA because they're bridging the gap here. And on one end, we have our 5' carbon here, and we have our phosphate attached there. And so this is the 5' end of this strand of DNA. And then we go down here, and we can see our 3' carbon right here is sticking off. And let me move this guy out of the way for now. And there's our thymine. And this is the 3' end, right? And we tend to follow directionality in the idea that we build nucleotides, right? We tack on new nucleotides to each of these, and we add them to the 3' end. So if this is my consistent strand that I already have, I'm going to put a new nucleotide right here. Okay? So I'm going to take the 5' end of a new nucleotide and attach it to the 3' end. So I always build from 5 to 3 in that direction. Okay, so that's an important concept. Now, if this strand of DNA was RNA, well, then we just tack on our hydroxy. Okay, so RNA is ribonucleic acid. There's the ribose. Okay, DNA is deoxyribonucleic acid. Okay, so we lose this. So this is RNA, and now I have lost that. It is now DNA. Okay, so that's the difference between DNA and RNA. Actually, I'm going to talk about that in the next slide as well. What's different, one other difference too, I'm going to mention this again in the next slide, is thymine becomes uracil if it's RNA. Now, it wouldn't be double-stranded like this, but so there is uracil. Okay, so RNA has uracil instead of thymine. That's literally the only difference. Well, it would, if it was RNA, it would also have the hydroxyl group. Okay. So that is now no longer thymine, that is now uracil. Okay, and all of these would have a hydroxy group on the 2' carbon, but it's kind of flipped. And so in our double-stranded DNA, okay, if it's double-stranded, it does not have to be, but most of the time we're going to be talking about double-stranded DNA. RNA is usually single-stranded. It's going to go in opposite directions. Okay, so we have our hydrogen bonding to hold the two strands together. We're going 5' to 3', 5' to 3'. Okay, in that direction, in this direction, and between our cytosine and guanine, so your C's and your G's, you have 3 hydrogens. Bonds typically, and between your thymine and adenine, or if you happen to be RNA, between your uracil and adenine, you can have 2 hydrogen bonds. So the more cytosine and guanine you have present in your DNA strands, the stronger the the stronger they are bound together. There's an extra hydrogen bond if you have more C's and G's. And so that can really add up and increase what we call your melting point of your DNA. So if you're trying to do PCR and replicate a bunch of DNA, knowing your C to G content versus your A to T content is actually really, really important. All right, so just to reiterate the difference between DNA and RNA, DNA is deoxyribose. You lose this hydroxyl group right here. That's really, really important. And then the only other real difference is the switching of uracil to thymine. Okay, so you can see the only difference is this methyl group here sticking off that carbon. And so otherwise, you're going to use all the same ones. Now, there's two kinds of bases, right? There are purines. Purines are really big. They have 2 rings. Okay, they have a 6 ring and a 5 ring, though they're stuck together, so it's technically sharing a couple carbons. And then you have your pyrimidines, which are a single hexagonal molecule. Okay, so purines are giant, pyrimidines are small. You just got to kind of know them. Now, if you're trying to string these things together to actually build a DNA molecule, which is the job of, or an RNA molecule, which is the job of polymerases. So there are DNA polymerases and there are RNA polymerases. Okay? When you're actually building from the 5' carbon to the 3' carbon, that's called a phosphodiester linkage. Okay, and it's a condensation reaction or a dehydration reaction where I'm going to take a water molecule. Water molecule is going to fall off here. Okay. And I'm going to build a new bond between the phosphate of my incoming nucleotide, and I'm going to attach that phosphate, which is on the 5' carbon, to the 3' carbon of this ribose. Okay. And that's how I build my DNA strand. That does have a positive delta G. Okay. So you have to have an enzyme do that, and it costs energy. You've got to link that together. The enzymes are going to make it easier to happen, but you're going to have to spend energy to actually build this, okay? Which is fine because actually storing information is really, really important. But it takes energy to store information, just like it takes energy to write down notes. It takes energy to store information. Now, to reiterate, so our primary structure would be just the order of nucleic acids. Okay. Your secondary structure is when we start, when we have 2 strands, okay? So this is different from proteins. So we spent a lot of time on talking about proteins, and primary structure is just the order of amino acids, okay? So that's the same for DNA's primary structure, right? It's just the order of nucleic acids. But DNA secondary structure is a little different, so we're going to take our 2 strands and stick them together. So we're just talking DNA here. So DNA secondary structure is this next level of structure where I have my hydrogen bonds holding these 2 strands together. I already mentioned G's and C's have 3, T's and A's have 2. Awesome, that's pretty straightforward, and the strength depends on that. Now, if we look at, we're going to zoom out again, okay? So once we start folding those on each other, that's our tertiary structure. So if you had just 2 strands next to each other, it looks relatively linear, it's kind of flat. That's still your secondary structure. But when I start twisting things around and forming my double helix, which is how we typically will view it, that is your tertiary structure. There are 2 grooves that show up. A lot of pictures of DNA are actually incorrect. DNA is a double helix, okay? So you actually have one strand rotating around the other, but they're both rotating, so it's a double helix. It's not a single helix. Let me pull up a picture of a single helix. So I pulled up here, I just did a quick Google search for DNA structure. And you can see this one is correct. This one looks fine. See, it looks kind of off, right? It's like a ladder that's bent. As opposed to, we go down here, this is not correct. So technology networks and naughty. This is a single helix. You can see the difference when we actually look at these twists. So look at this one. See, it's very, very even. This one is not, okay? So we actually have 2 grooves. It's a ladder that's bent, not a single 2 strands rotating, which it's hard to verbalize what I'm trying to say, but you just have 2 grooves, okay? And that's really, really important because there are lots and lots of enzymes or enzyme complexes that need to access DNA. And they want to access the nucleotides, okay? And so we have a nice wide gap, a major groove that allows access to the nucleotides. And then you have the minor groove on which other enzymes could maybe crawl along it, or maybe they don't need access to the nucleotides. They just want to move along the DNA, and they can hang out on the minor groove because they don't actually need access. And so the actual structure of DNA is very, very important. So that's your tertiary structure, okay? Tertiary structures vary. There are a few different kinds. So the B form of DNA, so that's this one in the middle, that is the most common form of DNA. Like in nature, it's usually B form. If you are dehydrated, so in humans not so much, but in bacteria we encounter this more. In your dehydrated form, you're actually kind of condensing it down. That's the A form. It is right-handed, so you can picture your handedness, which we encountered before with protein helix structures. You can be right-handed. It's kind of which way is the ladder curving if you use your finger. So and DNA tends to be right-handed. It's kind of curving towards your fingers. There can be left-handed forms though, so you can see that it's going in a different direction. Okay, and so the left hand, it's left-handed. The thumb is pointing up, as opposed to these ones. The thumb is kind of pointing down, or well, it would be pointing down if you twisted it. Whatever. Right-handed, left-handed. I never liked the idea of handedness, but whatever. It's twisting a different direction. Okay, this one is kind of like going up, and this one's kind of going down, but it depends on which angle you look at it. Whatever. Dehydrated, not dehydrated. Z form is weird, poorly understood. It probably has something to do. You can dig deeper on it. It's just not super well studied, and it's associated with a couple diseases. It turns out your cells could probably use the Z form to change the rate of transcription, but we're not going to focus on that. Let's talk about some other structures. DNA itself, so this would actually be tertiary structure. My bad. So you can have non-traditional hydrogen bonding. So let's talk about this. So traditional hydrogen bonding is called Watson-Crick bonding. Okay? That's the idea of having 3 hydrogen bonds between G's and C's and 2 hydrogen bonds between T's and A's based on their structure. It doesn't have to always be that way. It turns out it was predicted early on that you could have alternative hydrogen bonding, and then it was found later, like 20, 30 years later, when we had the technology to actually see it. And so you can get DNA to actually fold over. I know this is spelled wrong, Harry Pen, really. I know that. It's just the best picture that I can find. Of course, it has a spelling error. I guess I could fix it, but whatever. So you can get DNA to fold over on itself in weird ways because you can get the hydrogen bonds to behave differently. So Hugstein is the most common version of that. I'm trying to think, I can't even think off the top of my head if there's other versions, but this is the most common one that we've understood. And basically, you're getting your distance between your A's and T's is much shorter, and your C's and G's is much shorter. So they're shortened hydrogen bonds. Okay, so hydrogen bonds can have varying lengths. We've talked about that in a previous lecture with enzyme active sites changing the bond length of the hydrogen bond, which actually can increase or decrease the pKa based on how tightly you're sort of squishing that hydrogen towards the oxygen. The tighter and the shorter your hydrogen bonding, the less likely you can lose that hydrogen, right? It's stuck closer to the oxygen. And so it's harder to pull off. And that's the kind of thing that we see in Hugstein binding. So we have much shorter hydrogen bonds, which, and also we lose one of the hydrogen bonds between G's and C's, which you can kind of imagine if I take my DNA and I can squish it closer together and kind of bend them towards each other a little bit, I can get some weird bending patterns. And that's what we see. These quadruplexes, they're often called G quadruplexes. You see these a lot in certain DNA folding patterns. It happens inside your cells. Triplex DNA, not so much, but just weird shapes. DNA is a very dynamic structure. We're going to talk about that in a second. How it interacts with its histones, how it interacts with itself, how it interacts with other enzymes can depend on these kinds of structures, which is really, really interesting. There's some work that's gone into this. You can actually see, so you can see this is like an NMR looking at the actual peaks that show up between G's and different nucleotides. If you want to read this article and learn more about it, go for it. But they're just basically doing an NMR and then seeing the different peaks that show up. And you can see the different peaks that change. It has to do with studying homologous recombination, which I will mention in a little while near the end of this presentation, and just how we actually fix DNA strand breaks, DNA mutations, and it turns out if you have mutations in certain structures, they're either forming quadruplexes when they shouldn't, or they're not forming quadruplexes when they should, and that can be a problem or a good thing. And so you being able to adjust that using different enzymes is important. So that's what that comes from. If you want to dig deeper, the link is there. Okay, let's talk about tertiary. You could argue quaternary, but I'm just going to call it tertiary structure of DNA. So that's where we get sort of epigenetics and the actual folding of the folds, okay? And so if we have our DNA and we start to coil the DNA, so we have our DNA, right? It's got its tertiary structure, and then we start folding these over. So it's up for debate. I would actually probably call this quaternary. The reason we don't typically say that is because quaternary to nerds sort of implies that we're having two DNA pieces stuck together. I don't like to ask questions about these things because it kind of depends on who you ask what level of structure that is. So don't worry, I'm not going to ask you a question on that. But if we start folding things around, we can change the density of our DNA. But that means we need to talk about how DNA is actually structured. So we have our initial DNA, and we take our DNA strand and we fold it over on histones. So there are a variety of histones. Histones are proteins that are associated with DNA, and together we call that chromatin. So that's a classic question that you get asked in a variety of classes, like, what is chromatin? Well, that's your DNA plus your proteins. And that's arguably, the proteins are just as important as the DNA because you have to keep your DNA nice and organized. If there aren't histones to keep your DNA organized, it's going to fall apart, you can't access it properly, and you're not going to open and close it properly when you actually need to engage in transcription. Okay? As you start folding those folds, so you can see the DNA is in blue, it's wrapped around your histones, you start folding those on top of each other, and it gets really complicated and it gets really dense. And here we have a chromosome, and that's the chromatic structure. And this would have undergone S phase, okay, because we have two sister chromatids here. And it would be entering mitosis because mitosis is the only time when you actually have condensation like this. It wouldn't normally look like this. So that's how much you can tell just from the picture of it. So, but we're not talking about that. We're talking about the structure. So let's talk about how DNA is actually wrapped around its histones, a really interesting idea. So I mentioned earlier, the density of chromatin is actually affecting epigenetics. So let's talk about epigenetics. It gets talked about a lot in the news by people who probably shouldn't be talking about it. There's a sub-industry of epigenetics, like a lot of nutritional groups will talk about epigenetics, like changing your epigenome. And if you eat lots of special stuff that we sell in pill form, it changes your epigenome and makes you healthier. What does it even mean to be healthier? That's such a glittering generality. So let's talk about what epigenetics actually means. So histones are a variety of proteins. They're not one protein. There are a bunch of different ones. So I used to have more pictures of this. I actually have more structure here in this picture here. There's different histone proteins. There's ones here that are kind of like the core ones, and then there's one that actually interact with the DNA. They themselves have structures. So up here you can see, here's our histone. There's these kind of histone proteins. There's ones that act as like more structural, as not really interacting with the DNA as much. Your DNA is this weird string looking thing. And it turns out when I change, so these little grippy guys, so they're going to hold on to the DNA, and if they're grabbing it, that keeps the DNA from expanding out and moving out too much. And so it can hold it really tight. And it turns out that the tightness of our histones is really, really important. We call that our histone density, and that's what epigenetics is. So if I have my histones and I can condense them down and make them really tight, like this picture right here, we call that the closed off form. Okay, I like to just call it's dense chromatin versus decondensed or relaxed chromatin. There's hetero and euchromatin. I don't like those terms, so I'm not going to use them. It's either condensed down or it is loosened or relaxed. There's a million different terms that you can use for this, but basically it's really dense and you can't access the DNA. That's what the key here is. And so this would be a great way if you were like a neuron and you didn't want to express like an immune gene. Or you're a neuron and you don't want to express epithelial genes because you don't need them. Why would I express an epithelial protein if I'm a neuron? Um, or just whatever protein I don't want to express. And so why not take that gene? The gene would be contained within the DNA itself of these histones. And so why don't I condense down those histones? And so one way that we can do that is to deacetylate, because it turns out when I take acetyl groups and I stick them on the histones, that can open it up and make it work better. So you can see here I have histones that are acetylated, okay, by histone acetyltransferases. That's a whole set of enzymes that can add acetyl groups. I say acetyl and acetyl, I use them interchangeably. It's really weird. I'm just out of my mind. And so when you add acetyl groups, generally that tends to open up your chromatin. So if you deacetylate, that tends to close them off. And there's been a push lately, by lately I mean probably like in the last 5 or 10 years, to develop inhibitors for those. And so it turns out HIV, like if you get HIV, it embeds its DNA inside your DNA. And so let's say your cells actively replicating, whatever, HIV comes in, embeds its DNA in here, and then you close that DNA off, okay? Using a histone deacetylase, so it's taking the acetyl groups off, histone deacetylase complex, HDAC. And so it takes that off, and now it's nice and dense, and then the HIV is like hiding in there. And so the idea was, well, if we open up the chromatin, if we add hats or let hats do their thing, and we add acetyl groups, then it opens up and I can get that HIV DNA to start being made or be transcribing and making HIV proteins, and now I can see the cell. Like, oh no, that cell has HIV. Otherwise, you don't know. It's just DNA kind of hiding in there. And so that's where that comes from, and that's what this diagram is talking about. So if you use an HDAC inhibitor, it can't remove the acetyl groups, and so they get acetylated, and then you can see the DNA. The HIV DNA. Now, if acetyl groups tend to open the histones, then the opposite would, in general, be methylation. And so I apologize for the quality of this picture, but it was honestly one of the better ones I could find because it shows like fun each histone protein piece. And so if you're methylated, you tend to be dense and condensed down. Well, here's an example. So that's heterochromatin, which it looks different from the other chromatin, the euchromatin. It's such a stupid way to look at it. It can condense down if you add methyl groups. So you can deacetylate and add methyl groups, and that tends to make it nice and dense. If you acetylate and demethylate, that can kind of open it up. And so the balance between those things can sort of decide how tightly packed or how loose your chromatin is. And so next, I just want to show just a brief video that's going to talk a little bit more about epigenetics. Yeah, let's cut to that.
Here's a conundrum. Identical twins originate from the same DNA, so how can they turn out so different, even in traits that have a significant genetic component? For instance, why might one twin get heart disease at 55 while her sister runs marathons in perfect health? Nature versus nurture has a lot to do with it, but a deeper related answer can be found within something called epigenetics. That's the study of how DNA interacts with the multitude of smaller molecules found within cells, which can activate and deactivate genes. If you think of DNA as a recipe book, those molecules are largely what determine what gets cooked when. They aren't making any conscious choices themselves. Rather, their presence and concentration within cells makes the difference. So how does that work? Genes in DNA are expressed when they're read and transcribed into RNA, which is translated into proteins by structures called ribosomes. And proteins are much of what determines a cell's characteristics and function. Epigenetic changes can boost or interfere with the transcription of specific genes. The most common way interference happens is that DNA, or the proteins it's wrapped around, gets labeled with small chemical tags. The set of all of the chemical tags that are attached to the genome of a given cell is called the epigenome. Some of these, like a methyl group, inhibit gene expression by derailing the cellular transcription machinery or causing the DNA to coil more tightly, making it inaccessible. The gene is still there, but it's silent. Boosting transcription is essentially the opposite. Some chemical tags will unwind the DNA, making it easier to transcribe, which ramps up production of the associated protein. Epigenetic changes can survive cell division, which means that they could affect an organism for its entire life. Sometimes that's a good thing, Epigenetic changes are part of normal development. The cells in an embryo start with one master genome. As the cells divide, some genes are activated and others inhibited. Over time, through this epigenetic reprogramming, some cells develop into heart cells and others into liver cells. Each of the approximately 200 cell types in your body has essentially the same genome, but its own distinct epigenome. The epigenome also mediates a lifelong dialogue between genes and the environment. The chemical tags that turn genes on and off can be influenced by factors including diet, chemical exposure, and medication. The resulting epigenetic changes can eventually lead to disease if, for example, they turn off a gene that makes a tumor-suppressing protein. Environmentally induced epigenetic changes are part of the reason why genetically identical twins can grow up to have very different lives. As twins get older, their epigenomes diverge, affecting the way they age and their susceptibility to disease. Even social experiences can cause epigenetic changes. In one famous experiment, when mother rats weren't attentive enough to their pups, genes in the babies that help them manage stress were methylated and turned off. And it might not stop with that generation. Most epigenetic marks are erased when egg and sperm cells are formed. But now, researchers think that some of those imprints survive, passing those epigenetic traits on to the next generation. Your mother's or your father's experiences as a child or choices as adults could actually shape your own epigenome. But even though epigenetic changes are sticky, they're not necessarily permanent. A balanced lifestyle that includes a healthy diet, exercise, and avoiding exposure to contaminants may, in the long run, create a healthy epigenome. It's an exciting time to be studying this. Scientists are just beginning to understand how epigenetics could explain mechanisms of human development and aging, as well as the origins of cancer, heart disease, mental illness, addiction, and many other conditions. Meanwhile, new genome editing techniques are making it much easier to identify which epigenetic changes really matter for health and disease. Once we understand how our epigenome influences us, we might be able to influence it too.
So that is one of those nice TED Ed videos. I like TED Ed videos. They're really nice. A little simple, but hey, it's nice and they have good animations. So great. Let's move on. So we're going to start talking about RNA. So RNA can also have secondary structure. So if DNA secondary structure is when we have two strands that have hydrogen bonding in between them, RNA is very, very similar. So RNA, a lot of times we think of RNA as being this one big long strand of messenger RNA that we can then translate into a protein, but that's not entirely true. RNA has a ton of structure to it. RNA structures are incredibly complex. You can get bulge loops, multi-branch loops, all these folding patterns. Hairpin loops are probably one of the more common ones that you'll encounter. And it's just where, RNA is also nucleotides, and nucleotides can form hydrogen bonds. And so you can form really, really cool structures when you have hydrogen bonding in RNA, and it happens a lot. RNA is not just one big, long linear molecule, typically. I had a typo. It said tertiary, I meant secondary. So the folding over is really, really interesting. And you can get these nice, fun shapes. They can do a variety of things. I'm going to talk about that in a second. There's different kinds of RNA. Messenger RNA is the most common, but there are other ones. There's actually a ton of other ones. Transfer RNA is one of the more common ones that we'll see when we talk about translation. So that's where you have your RNA, lots of hairpin loops here to give it some structure. And then down here would be the anticodon that actually matches up with the messenger RNA. And then up here is where you attach your amino acid and can do translation, which is very, very fun. And then these are just different models of the structure. And so you can see this is RNA, but it has a ton of really cool structure to it. So that's pretty awesome. There are a bunch of different kinds. Here's a list of a variety of ones and based on size. So your messenger RNAs can be huge and massive. And that also includes ribosomal RNA, so that's rRNA, gets made inside the nucleolus, and it's huge. Those are structural components of ribosomes. Ribosomes are not just proteins. They are ribozymes. So they are proteins complexed with large pieces of RNA. You can have other smaller pieces of RNA. I want to talk about some of the functions of these. as well. Now, something that I had some experience with in grad school was using aptamers. Now, there's a lot going on here, but basically what you can do is if you get your RNA folded properly, it can interact with other things. It doesn't even matter. So you can hypothetically fold an RNA into an infinite number of structures that are stable. And so maybe infinite's the wrong word, but you can get them to fold and interact with things. So they can interact with proteins, they can interact with lipids, they can interact with other RNA or DNA molecules. And so what you do is you take your protein that, let's say it's a protein that you want to grab onto with RNA. And so you would take your protein and you put it in a column, and then you take millions of different RNAs all folded over in different ways, and you see which ones interact and actually bind to your protein. And it turns out you can use that as a drug delivery system. So we were doing that to deliver, so you can see here, this would be a surface receptor that would grab onto the aptamer. So that's what these are called. These are called aptamers, or the little RNAs that we use. And then on the other end of the RNA, we would attach it to something else. So like we were doing an anti-HIV drug, or you could do like a gene therapy if you want it really targeted. So you just make sure that you're targeting this specific receptor. And so it's just a way to make an RNA molecule that was really, really specific. Now, another idea is the idea of RNA interference. So RNA interference is a really, really cool concept. You'll see it a lot in primary articles where they'll use RNA interference to perform a knockdown of a specific gene. Like if you have a protein and you want to see what happens if I get rid of this protein in the cell, well, instead of going in and trying to delete the actual gene, all you have to do is throw in interfering RNA that's complementary, and you throw that in, and it will grab onto the actual messenger RNA for that protein, and it will cleave it. And so you're not making the proteins off of that. There's two big kinds. There's small interfering and there's micro. So small interfering is really, really specific, and that's oftentimes what you'll be using if you were in a lab and trying to do very targeted silencing. And so basically you just provide, using dicer, it cuts up the small interfering RNA complex. Ago gets it single-stranded, and then the RISC complex is what you actually use as like the weapon. And so RISC is going to use this new template. So this top piece here is the template, and then that grabs onto this messenger RNA, 3' to 5' here, and it will cleave it. MicroRNA is similar, but it doesn't require as much complementarity. So with small interfering RNAs, you need like the whole length of this to actually be complementary to the RNA you're trying to silence. MicroRNA is much less stringent, less stingy. So you could maybe only have like 2 or 3 or 7 nucleotides, as opposed to the entire length, which would be like 21 nucleotides long. And so you can knock out a bunch of different RNAs with only one microRNA. So it's a little broader, which is kind of cool. So I'm going to cut to a really, really well done Nature, like the journal Nature video that they have that shows some of the ways that we can use siRNA and how it actually functions inside of a cell.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future perhaps as a therapy. This animation will introduce you to the principles of RNAi involving 2 important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs or double-stranded RNA is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein. One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand. The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
So that is a great visual image of how RNA interference works. Like I said, super common in a lot of more modern experiments where they want to test and knock something down. So leaving DNA and RNA, I wanted to talk a little bit about some of the different aspects that molecules can have, that nucleic acids can have. So one of the big ones is energy storage, and ATP we talk about all the time, right? Adenosine triphosphate. Well, it's an adenosine that is a nucleic acid. And so if we tack some phosphates on there, normally if we just have like one phosphate, like an AMP, an adenosine monophosphate, that's kind of what we use to make our DNA strand, sort of. And so if I tack on more phosphates, the actual bonds between those phosphates are highly energetic and very unstable. They're stable enough that you can have a molecule, but they're unstable enough that they're pretty easy to break, which gives you a nice negative delta G. So we can couple that to other reactions, and then you can sort of string together a really positive delta G reaction with something that has enough negative delta G, and then you can actually get a reaction to occur or to become spontaneous. Now, there are two big ways that we make these. Well, there's really three ways to make ATP. Plants can make ATP, but the main way that we're going to be talking about in this class is through substrate level phosphorylation and oxidative phosphorylation. Okay, so substrate level happens during glycolysis, and that's where you physically, so here we have one, that's where you're physically pulling a phosphate off and sticking it onto ADP to make ATP. Okay, so that's substrate level phosphorylation. We're going to talk a lot more about that later on when we talk about glycolysis. Sorry. When we talk about the citric acid cycle, I actually want to talk about that next. Okay, so that's oxidative phosphorylation. So the citric acid cycle and the glycolysis sort of lead into the electron transport chain, where we put hydrogens on one side of a membrane and use an ATP synthase to, it takes the energy of having a nice concentration gradient of hydrogens to actually power the machinery that builds ATP, that takes a phosphate and tacks it onto ADP. And we're going to talk more about that later. But some of the molecules that we get out of the citric acid cycle and glycolysis, we can use to put hydrogens on the other side of the electron transport chain and create that gradient. And those are also nucleic acids. So we call these redox molecules often because we're reducing and oxidizing. And so in this case, reduction is the gain of electrons, right? So we're going to reduce FAD and NAD to FADH2 and NADH respectively. So, and that occurs more often in the citric acid cycle. You do get a few NADHs out of the glycolysis, but FADH2 and NADH, generally we get those out of the citric acid cycle. And then we're going to follow those to the electron transport chain. But structurally, they're really, really interesting. And the whole point of them is just like ATP. So if the point of ATP is to build this bond here to store energy, well, that's it's being reduced, right? I'm taking electrons and an atom, but I'm taking electrons and sort of storing them in here. And so we're going to do that with our flavin adenine dinucleotide, so that's FAD, as well as our nicotinamide adenine dinucleotide, okay? So there's your NAD, and so what you're doing is there's a lot of structure to it, just like ATP has a large structure to it. I'm actually going to show you a picture of ATP in a second. But ATP looks very, very similar to this. And on the top here, so you see this hydrogen right there? So this is our NAD plus. It's technically plus, so sorry. So there's our NAD plus, okay? And we're going to tack on an electron. It's going to balance out that positive charge, but we also give a hydrogen there. So the hydrogen is what's also holding on to that electron. And so that goes from NAD plus to NADH. And so you can see this, we have an extra hydrogen up here now. Okay? And so that is our reduced form. And reduction is a gain of electrons. So we're tacking electrons into this molecule, and then we're going to use that molecule because it's more stable, and we're going to transfer the molecule towards the electron transport chain. And then we're going to then rip those electrons off and send them into the transport chain. And in the process, this hydrogen is going to end up on the other side of that membrane. And we're going to get a ton of those over there. It's going to become super acidic, and then we're going to use that gradient. FAD, same thing, but the hydrogens get tacked on to right here and right here. And FAD is really, really cool because you can have FADH, not FADH2, but FADH. And so it allows you to, you don't have to have two electrons attached. You could have just one. And so you get a little bit of diversity there, which is nice. So you could tack a hydrogen here with an electron here. You could tack one here with an electron, or you could tack one to both sites. And that gives you some flexibility, is probably a better word than diversity, for actually using this molecule to move energy. Really, really cool. So let's take a look. So here's AMP down here. So there's one, or up here, one phosphate, two phosphates, three phosphates, monophosphate, diphosphate, triphosphate. There's your ATP, very, very typical. You can break this phosphate off and get a bunch of energy out. You can also break this phosphate off and get a decent amount of energy out. These are all negative delta G reactions when I pull those off. Now, there is something else that you can do, though. You can take AMP, okay, and you can reform this bond here. Okay, so we got this double bonded oxygen here. We can sort of have that fold back onto the ribose here and form a cyclic AMP. It's still adenosine monophosphate. Okay, it's still an adenine with a ribose, and we still have one phosphate here. I think I misspoke a little bit, but it's this bond here because you still have the carbonyl. But we're kind of reforming this guy down here. Yeah, that's what it is. Sorry. It's been a long day. So you would form a bond here and make it cyclic, which gives it just some different properties. It kind of makes it more stable, basically. So you're not able to pull this phosphate off as well. And so it becomes a messenger molecule. And so one of the functions that I mentioned earlier was signaling. So we've talked about information storage, we've talked about energy transfer like ATP, we talked about redox coenzymes, and now we need to talk about signaling. And so something that can come from signaling is using adenosine monophosphate in its cyclic form. There's a lot going on here. I realize that. I actually have two diagrams here. I actually have three. The next one is also a diagram. So what's going on here? We're going to focus on the left side here, which is from Khan Academy. So epinephrine, That's also adrenaline. So when you get a nice burst of adrenaline, it's your fight or flight, right? So you need to be ready to defend yourself from a wild bear or something. And so what you want is nice, readily accessed glucose to power your cells, right? You don't want it trapped inside some giant polymer. And so glycogen being the polymer, and you want glucose. So this is what you want to happen if you have epinephrine binding. So you have epinephrine binding to your epinephrine receptor, and that has a component that slides over and activates your adenyl cyclase. And that takes ATP, so you need to have ATP around, and turns it into cyclic AMP, okay, which can then go on and activate your glycogen phosphorylase. And your glycogen phosphorylase's job is to convert glycogen to glucose. And then you have glucose readily accessed, and you can do stuff with it. Is cyclic AMP is, it's a really common signaling molecule. And so what's happening, yes, you're turning on the glycogen phosphorylase that's going to convert glycogen to glucose. Awesome. But it also turns off the enzyme that makes glycogen. Because why would you be making this? To make it, you have to take glucose and make it into glycogen. And so you only want it going this direction, right? So we want this one turned on, and we want the one that goes the other way turned off. And that's what happens over here. So we have an inactive glycogen synthase, and that's driven both by cyclic AMP, which is really, really cool. This is just a more complicated diagram over here. It's almost the exact same thing. So there's cyclic AMP, and then it's activating a couple kinases, because eventually you want to phosphorylate glycogen phosphorylase to actually turn it on. And so that's what's happening down here. And the key to this one, I like this one because it's simpler to visualize, but I like this one because it emphasizes going from a single binding of epinephrine to the epinephrine receptor to the amplification, okay? Because cell signaling requires massive amplification, okay? And so I want to be converting all the glycogen I can to all the glucose that I can. And so it jumps up from 20-ish molecules of actual protein kinase initiated by the cyclic AMP, like right here, all the way up to 100 of the next one, 1,000 of the next one, and you're ending up with like 10,000 glucose molecules from the binding of one epinephrine molecule. And so that's really, really important. This is just another visualization of what's happening. Just to emphasize that the actual binding and movement of cell signaling is really, really complicated. When I teach cell biology, we talk about this a little bit more and go into more depth of actual signaling molecules. But the thing I actually wanted to emphasize was just, number one, that cyclic AMP is important, and that you can get amplification of your signal. And this one actually requires GTP, which is kind of fun. And it turns on to GTPase, and it moves over. It like physically slides over, which is really cool. There's the adenylate cyclase right here. Pretty cool. So if you wanted to sit and stare at this for hours, go for it. Now, cyclic AMP is not just associated with that cell signaling. It's a giant ubiquitous molecule. You see it everywhere. And so I'm an immunologist, and so just in the immune system, we see cyclic AMP having all of these, having some kind of association with all of these jobs, like B cells making antibodies, or neutrophils releasing stuff, or I don't like that it says histamine here. Neutrophils don't release histamine, but basophils and eosinophils can. Mostly actually mast cells, which are tied to basophil. Whatever, it's fine. But you can see they're just associated with lots of different things, and so that's kind of cool. Now you may be asking yourself, it's cyclic AMP. Is there like a cyclic GMP and a cyclic CMP and a cyclic TMP? Yes, we don't typically encounter cyclic TMP. I think hypothetically you can make it. As of right now, I couldn't find evidence of it actually working in a physical system. You sometimes will see GMP and CMP. GMP is associated with like muscles and some ion channels, which that kind of connects. Cyclic CMP, not as common. They've seen it in muscles and with some kinases, so they do act as signaling molecules. But cyclic AMP is definitely the major one. Moving on, in biochemistry, I wanted to emphasize this a little bit. If you take genetics, we will talk about this even more. But DNA damage and repair, if we're talking about nucleic acids, we need to talk at least a little bit about what happens when your information storage molecules actually become damaged. especially your DNA. So that is your master blueprint of everything inside that cell, right? And so if you get damage in a variety of different ways, well, we have a variety of different ways to repair those. And it's really, really important. And so I like this chart. It's nice. It's visual. I'm a very visual person. So if you have an insertion or a deletion, that's really bad because that's going to change what proteins you have. And then we use mismatch repair, which is a separate set of enzymes. If you have a single-stranded break, a double-stranded break, those are really, really bad. That's like X-rays can cause double-stranded breaks. What is this? Cross-linking, not as common, but you can have, basically, you can fix individual nucleotides, or you can fix big chunks of nucleotides, like 8 to 10 nucleotides long, or you can fix whole strand breaks through a couple different ways. Usually homologous recombination or non-homologous end joining, or NHEJ. That's for like the really bad things. And how do you decide what to do? Well, that's a good question. So let's get to that. I like this diagram a lot. I wanted it to have, I just wanted it in the slides so that if you ever needed to know even more detail about how these processes actually happen, then this one's really, really nice. It has all of the genes that code for these specific proteins that do these repairs, which is really, really cool. So this would be a great diagram to use for like an exam or something. Actual DNA repair, if you study the cell cycle, or if you've taken a class like genetics where we talk about the cell cycle, some of these should look really, really common. So when you're passing through the cell cycle, you go from G1 to S to G2, go through mitosis and over again. There's lots of checkpoints, right? Well, ATM and ATR show up in a lot of those checkpoints. BRCA, which is the breast cancer associated gene, there's BRCA1 and BRCA2, those are tumor suppressor genes. All of these are tied into a lot of the checkpoints during the cell cycle are looking for DNA damage, because you don't want to replicate a cell that has DNA damage, because that's really bad. It's not going to work right, and it could become cancerous. So that's really, really bad. And so we have tumor suppressor genes, and these are two of the big ones that look for specific damage to your nucleic acids, which is really, really cool. So non-homologous recombination, this is homologous recombination. Just one more thing I wanted to show you, because it has like a nice pathway. And so a great exam question would be to ask, if you don't have a CHK2, what kind of effect would that have on the cell? So like if you deleted this, what would happen? Well, it's not going to tell BRCA to do its thing, so you could get, I don't know, cancer or something like that. One more picture too, I just wanted to emphasize that a lot of the DNA damage, so here's DNA damage, right, occurring. So the DNA damage occurs, here's the actual damage, and then these are the ways that it gets repaired. But you may not want that to happen. So what if you have cancer and they're going to use like X-ray therapy or like radiation therapy? Well, that's DNA damage, right? And so what you may want to do is combination therapy. So you could have a patient who has a very accessible tumor. You could aim like X-rays at it really easily. Or like my grandpa had prostate cancer, and they gave him radioactive like seeds. They're like these little seed things. I mean, I was too young to understand what was going on, but they put these little seeds like in his prostate to release radiation and cause DNA damage. So you're basically trying to get the cancer cells to be the only ones that have single-stranded or double-stranded breaks, because those are really, really dangerous. And so the problem is those cells could repair themselves, and so you apply some kind of drug. And so if these non-homologous end joining is performed by BRCA1 or PARP1, which, lo and behold, PARP1 is associated with, that's this, PARP1. Oh, snap. Things are coming back. But if you block those using like platinum chemotherapy or PARP inhibitors, you would use these in conjunction with radioactive treatment. So that kind of makes sense. Like, if I'm trying to kill the cancer cell, I want to disable its ability to repair its own DNA. Last 4 slides here, I wanted to talk a little bit about some of the key experiments that were conducted to actually understand, basically understand DNA and how DNA it works inside the body. So first, we're going to talk, so there's 4 of these. So we're going to go through these, trying to go pretty fast. So think about the logic used here to determine these things. So what we didn't know, Hershey and Chase, they didn't know if DNA is what's transmitting genetics. Like what actually goes from parent to child? Like, is it protein based? Is it nucleic acid based? Well, we didn't know. And so, but what they did know was what those components were made of. So they knew that sulfur is only inside proteins. There's no sulfur associated with DNA. If you pull the DNA out and pull the proteins, if you separate the DNA, because we know DNA is very closely associated with proteins, right, with the histones. And so as long as you pull those apart, get rid of all the histones and separate those, and so you have all your proteins in one jar and all of your nucleic acids in another jar, you could label them. And so what they did was they took bacteriophages, which are just viruses that affect bacteria, and they grew them with radioactive sulfur. So all the sulfur that they could grow with, all the sulfur that they can use to make proteins is radioactive. And then they let it infect bacteria, and then they would grow the bacteria and then pellet it down. And so wherever that radiation is, well, we know that that's the genetic material, and the genetic material was not contained within the bacterial pellet. It was contained within the supernatant, which is where the phage was. Okay, so none of the protein was actually being transmitted to the bacteria. But if you label the bacteriophage's DNA and let it do its thing, well, it inserted its DNA into the bacteria. And so if you look at the bacteria in the pellet, it was glowing. Hopefully you were safe. Because they, instead of labeling sulfur, which is only going to follow the proteins, okay, so the bacteriophage is the radioactive, the actual outer coat. If you label it with phosphorus or phosphate, with phosphorus being the actual element, phosphate being what's actually in the, with a bunch of oxygens around it, same idea. So you have radioactive phosphorus, then you can track that and see, oh, okay, well, we know that the phosphorus was contained in the DNA, and that's what actually was transmitted into the bacteria. And so we know that's how viruses are transmitting their genetic material. Now, was that the only way? They didn't know at the time, but they just hypothesized that that's what was going on. Now, another experiment that was done was performed by Meselson and Stahl. It was really interesting. So they took two different isotopes of nitrogen, okay, N14 and N15, and those would have different densities. And so they let that grow in, they let their bacteria grow in the presence of N14, I believe. Let me double check. It was N15. They let it grow in the presence of N15. Duh, it would be heavier because it's N15. It has an extra, what is it? Neutron. And so that would weigh more. So they let it grow for one replicative cycle. So basically all their DNA got labeled. So in this case, these are all labeled like blue. And then they let it then grow subsequently in N14, which is a slightly lighter but still radioactive isotope. So you can still isolate both because they're both radioactive, but one weighs more than the other. And so when you do a density centrifugation, then they will separate out differently. And so what they wanted to know was they predicted that when you actually have DNA replication occur, that you pull each parent strand apart. So here's your parent strands. You pull those apart, strands, sorry. And then you build your new strand, and then those become the new daughter ones. And so you have half from the parent and half from the new strand. And so it's semi-conservative. So you've conserved like half the parental strand in each subsequent strand. And so that's what they predicted. But what they didn't know is they were like, well, I mean, we don't know that for sure. So half of them should have N14 and half of them should have N15. And then in the next generation, a quarter of them should have N14 and the other quarter should have N15 if you weigh it out, or the other 75%. So you can see it just kind of dilutes out the N14 or the N15 versus the N14. And so conservative would have been like, oh, okay, we pull apart these strands, build a new strand, and then put the two new strands together that are complementary, and then put the two parental strands that were together that matched up back again. I would argue that it could have been both of these. Now that we know how semi-conservative replication actually occurs, it seems really obvious. But back then, I mean, you never know. Like maybe the parent strands go back together. I don't know. We need to prove it, right? Dispersive would have been it's just like random. So you just like make a little chunk and another little chunk and another little chunk, and you just mix them all together, and it all gets mixed up, and it's just crazy. But if it was this way, then eventually, like 4 or 5 generations down the line, it would just be kind of all mixed together. Your N15 and your N14 wouldn't be able to separate out. But in this one, in semi-conservative, you actually were able to separate it out. So that's kind of cool. sets of scientists, and these are actually really, really closely related. So Griffith had an interesting idea. So he knew that certain bacteria would kill, I think it was staph, would kill a mouse. So you take it, but there were two different strains. So they knew, like, okay, this one strain is really dangerous. So that's the smooth strain. And what he was actually encountering was it was encapsulated. And so the smooth strain, you put it in the mouse, it dies. Okay, pretty easy. But the rough strain, which didn't have that nice capsule on the outside, you give it to the mouse, it's fine. Okay, cool. Well, what if I kill, like, with heat, so I, like, burn, sort of. You wouldn't burn it, but you would, like, heat it up so that you kill the smooth strain. Okay, well, I heat it up and it doesn't kill it, right? So it's not working. The bacteria is dead, right? But if I take these dead bacteria, mix them together for a couple days with this rough strain, what we now know is that the rough strain was taking up DNA from the smooth strain, either genomic or probably more likely plasmid DNA that gave it a virulence factor. And so now the only living strain is the kind that doesn't normally kill the mouse. But if you put dead versions of the really deadly strain with this one, it takes up some of that deadly strain's DNA that made it deadly, and then it kills the mouse. And so that was really interesting. So he showed basically transformation, that something was being transmitted between these two strains, and now we know it was DNA, to actually make it virulent. Double checked. It was pneumococcal bacteria. It was not staphylococcal. It was pneumococcal, which I should have known that because pneumococcal uses a nice capsule to be virulent. Now, kind of jumping off that, they wanted to confirm that this was true. And so what they didn't know was they're like, okay, we know something is being transferred. We predict that it's genetic, or that it's nucleic acids, but we don't know that for sure. And so Avery, McLeod, and McCarty, what they did was they did almost the exact same experiment, except they're not killing mice, but they are keeping track of whether or not new cells, so that's the 2R cells, are actually being able to, if they can actually take up, what material are they taking up? I'm sorry, I'm like doing a terrible job explaining this. So they took the cells and they spin them down, they heat kill them, so these are the bad ones, okay? These 3S ones are the bad ones. So they heat kill those, awesome. And then what they're gonna do is they're going to then take those cells and mix them with different things that will destroy different components. So they put it with a protease that'll destroy all the proteins. And then they took that and put it with other cells, the 2Rs, that don't, that could hypothetically, so the 2Rs would be these ones, the ones that don't kill the mice, okay? And so they're gonna take these, they get rid of all the proteins, but leave the DNA and the RNA, and it works, and the new cells potentially basically do this, they kill the mouse, okay? They're doing it a different way, but that's basically what's happening. So yes, the component that these bacteria that don't normally kill the mouse, the component that they're gaining from these heat kill bacteria is still present there, okay? Even though we got rid of the proteins. So therefore, we know the proteins aren't the ones transmitting that ability or that virulence factor. Okay, cool. So they said, all right, well, we know we have this enzyme that gets rid of RNA, which is just a different nucleic acid. So let's get rid of all the RNA. And what do you know? It still keeps happening, okay? And so we're like, okay, awesome. So we got rid of the RNA, still works. Oh, this was the control, sorry. So this one just, yes, it works. When we get rid of the protein, still works. When we get rid of the RNA, it still works. But guess what happens when you use deoxyribonuclease, which will destroy your DNA? It doesn't work anymore. So therefore, the factor that is being transmitted is DNA, which is kind of a cool experiment, but you have to think about what was involved here. They had to have these enzymes, right? And so as our technology increased, our ability to perform more and more complex and subtle experiments to kind of dig deep down got more and more complex. And that's where we're at now. We can do really complex experiments because those before us have paved the way.